my name's Georgina Gibson. Today I'm going to talk to you about how we can use models to understand the dynamics and early life history of marine organisms. I'm a researcher at the International Arctic Research Center, although I'm currently based down in Santa Fe, New Mexico. I'm still very actively involved with the community up in Fairbanks, so please feel free to reach out to me after this if you have any questions. My email right here, gagibson@alaska.edu. My goal for this lecture is to give you an appreciation for why we might use numerical models to understand early life histories, hopefully demystify models for you a little bit, give you an idea of what's involved in model development, and then empower you to ask probing questions either of your colleagues or as you read manuscripts that employ some of these modeling techniques that we're going to talk about. Models have a wide range of applications as research tools in marine science. They can be used to understand or observe phenomena and help us predict future patterns. They can be used to test hypotheses. We can use them to interpolate between the sparse data that we might have available. Compared to observational programs that use expensive ship time, they're relatively expensive to run. They're pretty easy to manipulate once you know what you're doing. And ultimately, model results can be used to help guide experimental efforts. Many of you may not be very familiar with modeling, so I'm going to ease you in with something that looks very familiar. And this is weather forecasts. So weather forecasters basically use atmospheric models and observational data to predict the weather at all times and places throughout the globe. These weather models will use data from various sources like satellites, Doppler radar, weather balloons, along with a series of equations that will describe what the atmosphere is likely to do based on how it looks now, and will allow us to get a weather forecast. This is very computation expensive, so we have large banks of supercomputers that do the equations for us. And we trust these weather forecasts so much that we actually base behavioral decisions on them. For example, we will look at the weather and maybe decide how you're going to dress or where you're going to go today. Marine ecosystem modeling is also essentially just a series of equations that we solve that describe how biological populations change with time. And with this kind of model, we can address questions like why is one region more productive than another, what controls a nuclear cycle in a system of interest, maybe is this going to be a more productive year, and what's going to happen to our ecosystem with climate change. So there's two types of ecosystem model that I want to introduce you to today. So the first we call an Eulerian model. So this is where we're observing or simulating dynamics of our ecosystem at a fixed location or grid point through which the fluid flows. So you can think of this as sitting at the end of a long jetty or pier, and you have your bucket and you sort of dip it in and you're periodically sampling the water and looking at its qualities. So you are not moving, but the water and whatever's in the water is moving past you. So this kind of model is simulating changes in average characteristics of a population. And a good example of this kind of model is what we refer to as a nutrient phytoplankton zooplankton or Mpz kind of model. We'll talk more about those later. So the other kind of model is what we call a Lagrangian model. So in this case, we're actually following individual particles or individuals as they move through space and time. So you can think about this as sitting in a rowboat that's just drifting along with the currents and then sampling water. So you are actually moving with the water or your individual. So this kind of model is good for organisms that have complex behaviors, and we refer to them usually as individual-based models or IBMs, although you might also sometimes hear them referred to as entity or agent-based models. The Eulerian or the population average style models don't usually work very well for fish or many invertebrates. The saying goes it's the unusual individual that survives and the average is dead, which is why it's important for us to follow the behavior and movement of a large number of individuals over time throughout our environment in order to understand where and what conditions are good for survival for the individuals. Individual-based models can work particularly well for modeling the early life history of many marine organisms, particularly those that have planktonic life stages. These individual-based models are usually coupled to a Eulerian style three-dimensional hydrographic model, which generates the currents, the temperatures, the salinity, etc., through which these individuals are moving. So I want to show you a quick example of some model output from an individual-based model to give you an idea of what we're talking about here. So let me just hit play. So in this example, each of the little black dots that you see moving around there represents an individual polyp larvae that we're simulating, and we're following the individual's behavior, growth, etc. as they are evicted around the Gulf of Alaska. So some of these individuals may be deemed successful in that they start from their spawning location. They maybe move through an area that has a lot of food, for example an ice edge algal bloom, so they get lots of food to eat, and then they end up in a settlement area that has the right kind of habitat for them. Whereas another individual may move from its spawning area. Due to advection, it might entirely miss the ice algae bloom. Then maybe it runs into a predator, so is subjected to predation, and it ends up at an undesirable settlement area. So these IBM style Lagrangian models are particularly useful for understanding these kinds of different environmental impacts that may be working on an individual. So although we're primarily interested here in early life history models, it's important to have an understanding of hydrodynamic models because they form the basis for the early life history models. They predict the currents, the velocities, diffuse and advect the individuals around. They produce the salinity, temperature, density fields that are important for understanding our larval and egg movements. So having an appropriate hydrodynamic model is critical to the success of your individual-based models. So I'm just going to spend a little bit of time giving you a brief understanding of hydrodynamic models. So the dynamics of the ocean is essentially described by the equations of motion and the equations of continuity. So if you're taking a physical oceanography class, you're going to hear a lot more about these equations and what goes into them. But even if you're not, it's good for you to at least have an awareness of the importance of these equations. So the equations of motion are derived from Newton's second law of motion, which you might well have heard of, force equals mass times acceleration. So if we apply this to oceanography and rearrange, we get the acceleration is, and our forces are pressure, Coriolis force, which is due to the fact that the Earth is spinning, gravity, frictional and tidal forces divided by mass. So if we apply this acceleration equation to our parcel of water we see here, you can see that we need to basically figure out the acceleration in each of these x, y, and z directions. So we end up with three equations, one for the acceleration in the x direction, one for the acceleration in the y direction, and one for the acceleration in the z direction. The other equation of importance is the continuity equation, which basically assumes that seawater is incompressible, meaning that if we were to change the velocity in one direction, for example the x direction, then we're going to affect a change in one of the other dimensions, for example the y or the z dimension. So now we have four equations and four unknown variables, u, v, w for our velocities in each direction, and p for pressure. So with four equations and four unknowns, the equations can be solved and the state of the ocean can be determined. Solving these equations by hand, particularly over a large model domain, would be extremely difficult. That's why we use a hydrodynamic model to do the job for us. So the equations are solved at every grid point throughout your model domain. Let me just pull up that. So this schematic just shows you a series of grid points that we've set up throughout our model domain, and at every one of these red dots the equations are being solved by the model. With that information we can then determine what the current system would look like within our model domain. So given that nutrient phytoplankton, zooplankton or Mpz style models often enable us to simulate the food availability for our early life history models, I want to give you a brief overview of this kind of model as well. So in an Mpz model, we usually have some kind of phytoplankton group, a zooplankton group, and some nutrients. So this is a very simplified depiction of an Mpz model with one phytoplankton group, one zooplankton group, and one nutrient group. And each of these groups are also acted on by the physical processes that advect and diffuse them around our model grid. So as with our hydrodynamic equations to describe the physical processes in the ocean, we also develop a series of equations to describe how our biological components in our model change with time. So for example, if we're looking at phytoplankton, the change in phytoplankton with time would be dependent on some growth. Then we'd see losses due to natural mortality, due to grazing by the zooplankton, and then we'd also see changes due to physical processes. So there's a series of equations that are developed basically to describe the changes that we would see behind each of these categories here. And these equations would be applied at every one of the model grid cells. So just as the physical model would apply each equation at each one of these red dots throughout our model domain. So in reality, many of the Mpz models that are used these days are not our simple Mpz model. Instead, we have many N's, multiple nutrients, nitrate, iron, ammonium, for example, multiple categories of the phytoplankton, for example, small and large, and then multiple categories of the zooplankton. So in this case, we have microzooplankton, small and large copepods, euphorsids, and even jellyfish. Of course, for every additional component you add, you have to come up with an additional series of equations and additional parameters that describe the behavior of your new component. For example, growth rates, mortality rates, respiration rates, etc. So with this added complexity, things get a lot more complicated and more computationally expensive. So to give you an example of some output from an Mpz style model, it's going to show you a quick movie. This is euphorsid concentration over a year in the Bering Sea. So let's see if we can get it going here. So this was simulated on about a 4-kilometer resolution hydrodynamic model, and then we had equations that described our nutrient, phytoplankton, zooplankton concentrations, and this is what we simulate the euphorsids to be doing. So that you can imagine with this information, you can then add in your individual larvae, for example, and see how much food might be available to them at certain places and certain times. There's a number of considerations that are important to think about when you're selecting your hydrodynamic model in terms of its appropriateness for your individual based model of your early life history. For example, make sure that it covers your domain of interest adequately. Is it simulating the key processes that you think are important for your individuals? For example, does it have the right transport mechanisms, tides, wind-driven circulation, freshwater buoyancy? Not all of these things are important to all individuals, but know your individual and what you think might be important for it. Also, does it get the temperature correct? Temperature can be very important for growth rates, for example. And are you simulating the right food source, if that's important for your individual? Also, does it have the appropriate resolution? And we're going to talk more about that in the next slide. So it's important to have a model that has a resolution fine enough to resolve your features of interest. However, as you get finer and finer in your resolution, models get computationally more expensive. So we need to find a trade-off. If we look at this global climate model, which has a horizontal resolution of about 2 degrees, you can see, if we look over the Bering Sea, while it's getting our temperature gradients about right, it's not really resolving any mesoscale features such as eddies that we know are there. Now, if we simulate this Bering Sea region with a finer resolution model, a 10-kilometer resolution model of the Bering Sea, we now are able to capture some of these mesoscale features. If we focus in here on one of these eddies, this red lines show the grid spacing for the 2-degree resolution model. And as you can see, we really only have one grid point that would fall within that eddy, so that's why you're not able to resolve this finer scale feature. Whereas with our 10-kilometer model, we have many grid points that fall within it, so we're able to at least somewhat adequately capture mesoscale features and their behavior. To give you more appreciation for the importance of resolution, I want to show you this image. Right now it's a really coarse resolution. I'm wondering if you can guess which famous person this is. Can't really tell, right, because it's not adequately resolved. If I was to simulate the same image at a finer resolution, it might become more apparent. Maybe some of you already have an idea who you think this person is. If we go finer again, it becomes even more apparent. And then we look at the full resolution, and it was Marilyn. Now, knowing what important features are, for example, Marilyn's hair or her red lips, if we go back one stage, for many applications we might decide that this resolution, for example, is appropriate enough and saves us a lot of computational power than simulating Marilyn at full resolution. Now let's talk about the number of particles you might have within your individual base model. So a single particle trajectory could be considered one trial of a statistical ensemble, but we're interested in the ensemble average behavior. So let's look at what one trajectory might look like. So for here example, I was releasing individuals over a seamount in the Gulf of Alaska, and I see that it ended up south of Kodiak Island. Now that's one single trial, and it might not be very representative of the population's behavior. So we run another individual, and we see that that one actually goes out into the middle of the Gulf of Alaska. So obviously we need to have more than 2 individuals in order for us to adequately resolve the population behavior. Running many individuals, we see that indeed the average behavior is to travel from the seamount towards the coast and settle somewhere along the coastal Gulf of Alaska. However, having too many particles can be computationally expensive, so it's important for us to find the balance of the correct number of individuals in order to resolve your behavior, but not too many that such that you don't have adequate computer power in order to run your model. Individual-based models have a wide range of complexity, depending on why you're using it or how much information you have about your organism of interest. So starting from the least complex, we could assume that an individual, for example our larvae, are neutrally buoyant passive particles that are simply advected and diffused around our system. Adding complexity, we might then give our individuals vertical and horizontal movement behaviors. For example, maybe add in some diel vertical migration. Getting more complex again, our individual particles could have metabolic rates applied. For example, growth rates, grazing rates, mortality rates, etc. And yet more complexity will be to add in predation pressures upon our individuals. So it's always good to start simple, but don't be afraid to add complexity when and where required. So before we start the actual sort of nitty-gritty coding of our individual base model, it's important to spend some time truly conceptualizing what we want the model to look like and what we want it to represent. So this important step will help us organize our thinking, make sure that the model is actually going to meet our stated objectives, ensure that you're going to actually simulate all relevant biophysical aspects of the system. You can identify gaps that may exist between the data that you deem necessary in order to build this model and what's currently available. You'll have to make decisions about your system boundaries, like we don't want to represent the whole world usually, maybe just a finer region. What is your target species? What are the key variables that are going to be in the model? Which life stages are you going to do? Often, as our individual gets older, it gets more complex movement behaviors, which maybe are too difficult for you to represent with this kind of model, for example. Or maybe we're only interested in the very early life stages of an individual species, for example. What is your model currency going to be? So often we'll track carbon or biomass, but maybe you're also tracking age or size of your individuals. What is your time and space scale going to be? And what are the important environmental variables that drive your individuals? This is an example of a conceptual model that I developed for a early life history model of sablefish that I have worked on. So before I really sort of started coding and developing my equations, I spent some time thinking about what I knew about sablefish and how I could represent them within a model. So for example, we knew that the adult sablefish spawn, releasing their eggs in deep water off the continental shelf in the Gulf of Alaska. Literature suggests that the eggs sink even deeper. They stay down there until they hatch into yolk sac larvae, at which point they begin to sort of migrate up into the water column, where they become feeding larvae, and you can see that they're growing all this time. I'm expecting my feeding larvae to maybe be up to about 34 millimeters standard length. At some point they transition into epipelagic juvenile larvae, and eventually they move onshore and settle in shallow nursery habitats in the Gulf of Alaska. So with this information, I knew that my hydrographic model had to cover most of the Gulf of Alaska, because I had to go from deep offshore spawning sites to inshore nursery sites. And I knew that I had to capture the finer scale mesoscale processes that were able to transport the sablefish on shore. I didn't want to simulate adult sablefish, so I decided that my first life stage that I simulated was going to be eggs. And so then I had 5 life stages with the eggs, the yolk sac larvae, the feeding larvae, the epipelagic juveniles, and my settled juveniles. I only followed each individual from its spawning time as an egg until it successfully settled in a nursery area. So as we continue to develop our IBM, we need to find ways of adequately simulating each of the processes important to your individual. For example, how are you going to simulate spawning? How are you going to simulate egg development, consumption, growth rate, mortality? And for each of these processes, we essentially need an equation that describes the changes we'll see in our individual. And so for this, I would look to the literature and to data in order to develop functional relationships to describe each of the processes you deem are important enough to include within your models. Sometimes you might find that such literature doesn't exist for your particular species, in which case we might need to borrow some equations and behaviors from similar species and see if using these equations you can adequately simulate the behavior of the population that you're interested in. So once we've fully conceptualized our model, the next step before actually developing the model code is to develop what we call pseudo code. So here we sketch out exactly what we want our model program to do for us. So at the start, for example, we're going to initialize all of our individuals. Then we're going to loop over each day within the model. And so at every day or time step, we're going to pull in the new prey field. We're going to pull in the new environmental variables. And then we're going to loop over individuals. So for every individual, we're then going to do the next set of code, which we have here. So we're going to look up its current depth, for example, and then project what its new depth is going to be and what its new location is going to be at the next time step. And then depending on which life stage our individuals are currently in, there's a series of equations that we're then going to have to solve. And we're going to have to decide at what point an individual is transitioning to the next life stage and then link to that relevant piece of code. So this is what pseudocode looks like, and then you can get stuck in developing the actual code to make this happen. So there's 2 main types of study that IBMs are generally used for. The first is what we call a connectivity study, and these look at how different spawning and nursery areas are related, for example, or where do the spawning products from different areas end up, or some spawning or nursery areas more successful than others? Do some areas fail to produce successful recruits? And the other kind of study that we might use an IBM for is what we call a longitudinal study. So in this kind of study, we look at the life history of an individual along its track as it moves from where it initialized it, i.e. where it spawned, and to where it settled. And this will help us understand more fully why some individuals are more successful than others. So now I'm going to finish by showing you a couple examples of those different kinds of study. So the sablefish model that I introduced you to earlier, I used with a connectivity analysis study in order to determine which spawning sites in the offshore Gulf of Alaska were most likely to produce successful settlers that made it all the way onshore and settled in a shallow nursery habitat. So in this, for this experiment, I divided the Gulf of Alaska into 12 alongshore regions, and I released individual particles that represented my sablefish in each of those regions. So over on the right is the actual model interface that I was using, and zoomed in here sort of gives you an example of my individual egg particles, and I released about 16,000 eggs per simulation. I ran the model for a year, and then I looked at which of these 12 alongshore areas particles had settled in. To summarize the results of a connectivity study, we developed what's called a connectivity matrix, which essentially describes the probability of an individual or larvae making the transition from its source, or where we spawned it, to its destination, or where it ended up settling, for example. So in my sablefish example, we released these individuals in the 12 alongshore regions, which are represented here on the y-axis, 1 through 12, and then I tracked the probability of them settling, again in the alongshore areas, but this time in the nearshore nursery areas. So what this matrix shows us, where we have this red hot color, is that spawning in areas 2 through 4, so over here, it has the highest probability of resulting in successful settlement in a nearshore area, and those nearshore areas are most likely to be 5 and 6, so sort of central Gulf of Alaska. This particular model didn't have any actual behaviors that would allow the individual to seek a location, so their transport was purely due to their vertical positioning within the water column, which individuals did control, and due to the advection by the physics in the hydrodynamic model. So this particular model experiment indicates that if individuals were released throughout the Gulf of Alaska, it's those that were released in the southeast that were most likely to result in successful recruitment of the sablefish, and they're more likely to settle within the central Gulf of Alaska. Now we actually do find individual sablefish settled over here in the southeast, and our model indicates that due to the physics alone, this is not the most probable scenario, which indicates to us that there's probably a behavioral mechanism that's important in sablefish that we're not currently representing within the model. So now let's take a look at examples of outputs from a trajectory analysis type individual-based model study. So this is taken from a project that was headed by Ken Coyle. Here he was looking at why different scenarios led to more or less successful copepods within the Gulf of Alaska. So he found that individuals that were retained on the Gulf of Alaska shelf, here for example, generally experienced trajectories that had the most production within them. The next most successful individuals tended to be those that were trapped within eddies, and which tend to be nutrient-rich, more productive environments than the offshore Gulf of Alaska, and individuals that were transported off of the shelf generally didn't experience very much production and were not successful. So these results indicate that the variability in transport, and thus the resulting trajectory of the individual particles of Neocalanus, can have very large consequences for secondary production in the Gulf of Alaska. So the individual-based models we've talked about today have been applied to a wide range of studies, including recruitment of marine fish and shellfish. They've been used to look at biological physical interactions that affect the early life stages. They've been used to understand which are the important mechanisms and processes for an individual. They've been used to examine the stock structure and connectivity of important species. They've been used to look at spread of invasive species. You can use them to look at the potential effects of climate change on recruitment. And lastly, they have been used to help design marine protected areas. So given all these real-world applications for our individual base models, it's important to understand the degree of trust that we have in models' output. Ideally, only a very thoroughly validated hydrodynamic model would be used as the basis for your particle tracking model. Validating a physical or a lower trophic level model is relatively doable given the availability of shipboard sampling and satellite data that is currently available. However, validating an individual base model is a lot more complicated. To validate a trajectory study, ideally we would be able to validate the passive component of the trajectory as well as its active or behavioral component. Validation could include some combination of acoustics, gliders, tracking released wax particles within the environment, or maybe looking at satellite track drifter outputs, as well as comparing modeled growth to laboratory-based growth studies, for example. If we were going to validate the connectivity kind of study, we could perhaps use tagging or mass marking data, as well as looking at genetic analysis to determine genetic structures of populations. So I'm going to wrap up today by leaving you with a few recommendations for if you're considering this kind of approach to your research. So start by defining the problem and the hypothesis. Are individual based models even the best tool? Why? Don't forget to start simple, but don't be afraid of complexity. Models should always be as simple as possible, but as complex as necessary. Best modeling practices depend upon the objective of the modeling exercise. There is no single model that's appropriate for all applications. Don't forget you may need to use data or relationships from different but related species, because not all data that you want is going to be available. And think in terms of model experiments. Don't just try to replicate nature. You'll find that by designing some well thought out experiments, you can often learn a lot more about the behavior of your population and species. Ultimately, recruitment dynamics is a very multidisciplinary problem, so try to work in multidisciplinary teams. Team up with oceanographers and ecologists. I hope you found this introduction to marine ecosystem modeling useful and have an appreciation of why we might use an individual based type model for understanding early life history dynamics. As I mentioned previously, I am a researcher with the University of Alaska, so if you have any questions about this work presented here today, or if you're wondering how you can include a modeling component in your own research, please feel free to reach out to me at the email above.